to Franco Izzo. And Franco is our second speaker today. And he's in the London laboratory at the Weill Cornell Medicine and the New York Genome Center. And he's an instructor in molecular biology medicine at Weill Cornell Medicine. And his research focuses on clonal hematopoiesis and myeloproliferative neoplasms. And the title of his talk today is Mapping Genotypes to Phenotypes through Single-Cell Resolution, Multi-Modal Approaches. So Franco, feel free to start whenever you are ready.
All right, thank you so much for that kind introduction. So basically in the lab, we're focusing on trying to develop technologies that allow us to capture both genotypes as well as another feature from single cells. And this effort started because of the recent realization that we are actually all genetic mosaics. And this is a very exciting frontier in human genetics. And we know that several studies have shown that there are pervasive clonal expansion across healthy tissues that are driven basically by somatic mutations. And this observation prompts what we think is a critical question, that is, what are the phenotypes that are driven by these somatic mutations? So I'm going to present two different works, two different technologies basically that we have developed. For the first one, we focus on the clonal expansion that occurs in hematopoiesis, or basically a term clonal hematopoiesis. And importantly, in clonal hematopoiesis, patients show normal peripheral blood counts, but they do have an increased risk of progressing towards an hematological malignancy. And for exploring this particular state is challenging to basically resolve the admixture of wild type and mutant cells that are present within each sample. So we know that single-cell sequencing allows for resolving the heterogeneity of cellular phenotypes, and the tools are really good at doing this. However, sometimes the phenotypes driven by these mutations are not strong enough to be picked up by these technologies. And then we need to actually link the genotypes to the phenotypes to be able to make comparisons between wild type and mutant cells present within a human sample. So the key question that we're trying to answer is, how can we capture single-cell genotypes and link them to cellular phenotypes? And our lab, as well as others, have developed methodologies that allow for a targeted genotyping in conjunction with single-cell technologies to try to address the impact of somatic mutations in humans. One of these first technologies is genotyping of transcriptomes, or GOT, which enables basically a joint genotyping together with single-cell RNA through targeted amplification of the site of interest in the transcriptome. So this is based on the 10x technology, and it basically implies the droplet capture of the cell together with a barcoded PID that allows barcoding of the transcripts and then to map them back to the single cell of origin. So what we did here was to include specific primers that amplify the locus of interest and allow to enrich for the transcript that contains the mutation. So this method basically leverages the expression level to try to map the mutation back to the cell. So in one of the recent projects, we apply this technology in particularly to clonal hematopoiesis, focusing on de novo methyltransferase 3A, or DNMT3A. And we focus on this variant in particular because while clonal hematopoiesis shows several different mutations, the R82 mutations in DNMT3A are more frequently observed in acute myeloid leukemia. So they're most likely involved in the evolution of the disease. So to overcome the scarcity of clonal hematopoiesis samples, because since the individuals don't have any blood abnormality, there is no clinically requirement for bone marrow biopsy, we scanned through a cohort of 130 autologous stem cell products harvested from patients with multiple myeloma remission, and we identified 4 cases with a high mutation fraction that allow us to use this genotyping of transcriptomes in these samples. And despite the low expression of DNMT3, we capture genotyping information for around 20% of the cells. So in this UMA, we see the cell types, and also on the UMA on the right, you see the projection of the single cell genotypes within this differentiation landscape. And you can see that the cells are intermingled between wild type mutants, which kind of underscores the requirement for a simultaneous capture of single cell genotypes and transcriptional information to try to understand what the impact of the NOD3A mutations are in this system. So despite being intermingled, we can already see some differentiation biases occurring in these samples. For example, when we focus on the myeloid versus lymphoid differentiation fork, we observe that mutated cells have a strong myeloid bias, and this is consistent with the strong association of this mutation in myeloid rather than lymphoid malignancies. So, for example, we can also take a step back and try to identify the differentiation biases more broadly. And for that, we calculated the normalized mutant cell ratio for each cell cluster, so for each cell type, which allows to chart the mutant fraction in a cell type dependent manner. Surprisingly, here you can see that in human clonal hematopoiesis, the mutant cell ratio is higher in immature myeloid progenitors, or IMPs, with the earliest in comparison with the earliest hematopoietic stem cell cluster, or HSPC. And interestingly, the immature myeloid progenitor clusters show an increase in the cell cycle gene expression, particularly in mutant progenitors. So this suggests that the mutation is providing a fitness advantage, specifically in the context of this particular cell state. So an additional approach that we took to look more into these differentiation biases is to compare wild type versus mutant differentiation probabilities based on RNA velocity. So here we found that the transition of mutant IMPs towards a megakaryocytic or erythroid fate is higher compared to wild type counterparts, which is with the opposite effect that we see in the probability of differentiation towards a granulocyte monocyte fate. So, and this recapitulates the previous observation from a lab using mouse models. But an additional advantage of having both wild type and mutant genotypes within cell types is that we can do comparisons within cell clusters. So, for example, we can do differential gene expression within the HSPC cluster within wild type and mutant cells and try to identify factors that can be involved in driving these differentiation skews. So, for example, when we look at transcription factors that could potentially regulate the genes that are differentially expressed, we observe, for example, MYC and MAX, as well as pro-inflammatory transcription factors such as TNF-kappa B and STATs that are involved in the regulation of these genes. However, we know that from transcription alone, it's hard to infer the transcription factor activity. So, especially we have no methylation information. So here we apply an additional technology in which we used a plate-based multi-modal single-cell platform that can capture DNA methylation, single-cell RNA, as well as targeted genotyping. So when we apply this, we perform differential methylation analysis, and we found a particular motif sequence that is enriched in the hypomethylated sites of the mutant cells compared to the hypermethylated sites. And this could be driving somehow the particular effects that we see on MYC. So to ask what this motif was, whether this motif was involved somehow in the phenotype that we're observing, we compared the similarity between this hypomethylated motif and the DNA binding motifs of the expressed transcription factors in this system. And here we can see, for example, that MYC and MIB, and one of the most similar motifs compared to the hypomethylated motifs that are expressed in the system. So this observation suggests that specific TFs may be preferentially affected by this mutation and provides a potential link between the methylation changes and specific phenotypic changes. So something else we can do is to actually look at the predicted MYC binding sites and look at the methylation status. We actually saw hypomethylation of these sites, and this hypomethylation was associated with increased transcription of the targets. However, while this technology is really useful to account for genotypes of highly expressed genes in particular, we know that this depends on the expression level of the target as well as the distance between the 3' and the mutation site because of the poly A capture that is used in the tenex. And we saw this back in our publication in 2019. So when we applied GOT for CALR, for example, which is a highly expressed gene, we were able to genotype 85% of the cells. When we tried the same approach for JAK2, which is a low expressed gene, and the distance from the gene end is above 2 kb, you can see that the genotype efficiency decreased quite drastically. So only 7% of the samples were actually captured. So how can we overcome these limitations in GOT, which are related with expression and distance from the gene end? And for that, we recently developed genotyping of targeted loci with single-cell chromatin accessibility, GOTCHA for short, which is easy. And this is basically using the 10x approach for single-cell ATAC-seq. And the main advantage of this approach is that it basically generates the genomic capture in droplet and targeting directly the genomic DNA. So to go through this quickly, we first do nuclear isolation and transposition, as you do in a standard ATAC-seq. But during the droplet generation, we flow into locus-specific GOTCHA primers that here in step 4 allowed for simultaneous capturing of chromatin accessibility, but as well as amplification of the targeted genomic region for which we want to capture the mutation site. And importantly, the primers are designed in a way that while the ATAC library is amplified linearly, the genotyping library is amplified exponentially. So we have a relative enrichment of the GOTCHA amplicons within these libraries. And then in step 5, we just split and do the library preparation of both ATAC and the GOTCHA genotyping library. And then we can basically map back the genotypes using the shared cellular barcodes. Just as for every new method, we started doing a cell mixing experiment, trying to benchmark our accuracy and our efficiency. For this, we did a cell mixing study with 2 human cell lines with distinct genotypes. As you can see here, we can identify both cell lines based only on the accessibility profiles, and we can verify that these cell lines are actually distinct based on both gene markers and the presence of mutually exclusive mitochondrial variants. But most importantly, when we map back our genotypes obtained by GOTCHA, we can clearly see that we have a high accuracy of 99.7% of the cell's genotype and 50% of cell capture. Importantly, we wanted to validate this, so we knew that wild-type cell line had an amplification in chromosome 9. So based on GOTCHA genotype labels, we split the cells and we perform copy number variation inference from the ATAC-seq data, and we indeed saw that the wild-type cell, as called by GOTCHA, contains this chromosomal amplification. So the next question was whether we could capture heterozygosity using this method. So we performed a second cell mixing experiment, this time including also a heterozygous cell line. And we saw that first, the homozygous genotyping was consistently regarding accuracy and efficiency with the previous experiment. And for the heterozygous cell line, we found that around 35% of the genotype cells basically exhibits successful multi-allelic capture. So GOTCHA is able to detect heterozygosity to a large extent. Something else we noticed in this particular experiment is that the cells recovered were not even across the different cell lines. And we realized that the percent of cells genotype was directly correlated with the copy number of the targeted site, which makes sense. So the initial input for the capture is higher, so the probability of capturing a genotype for a given cell is also increased. But most importantly, the accuracy was high and remained high across all the different, from copy number of 1 to 7, it remained above 99%. So after doing this, we moved to applying GOTCHA to human samples to try to define epigenetic changes that are driven by these somatic mutations. For this particular project, we selected myeloproliferative neoplasms as our disease, basically because these diseases show an increased proliferative drive of mature myeloid cells in the bone marrow. There's a risk of a spent phase that is characterized by bone marrow fibrosis, and there's a high risk of transformation to acute leukemia. But most importantly, all these diseases have a constitutively activated tyrosine kinases. And in particular, primary myelofibrosis, essential thrombocythemia, and polycythemia vera are driven mainly by mutation in JAK2, which is the gene that we intended to capture with using GOT before, and we only capture 7% of the cells. So we apply gotcha to focusing on primary myelofibrosis, and what we saw is that first we have high quality ATAC-seq data, so we can I'm showing 7 patients integrated in a single HuMap, and we can confidently identify the cell types that are involved and they're expected in hematopoiesis. But most importantly, when we map back the genotypes obtained by gotcha, we can see that we obtain genotype for 16,000 cells, which is just under 50% of the cells genotype, compared to and then here you can see on the right that compared with the previous studies using transcription-based genotyping, this is almost a 5-fold increase in the efficiency of genotype capture using this method. So as an example of the things we can do with this kind of data is we can first look at the, of course, mutant frequency across the different clusters. And here we notice that in the samples, in particular in the untreated samples, so we have here patients that are not under treatment and patients that were treated with ruxolitinib, which is a JAK2 inhibitor. So here you can see that in the treated patients, you have an increase of the mutant fraction along the differentiation trajectory towards erythroid or macrocytic phase, which is a known bias in these diseases. But the interesting thing is that when we look into patients that were treated with ruxolitinib, this differentiation bias seems to be basically abrogated, and there was a more homogeneous distribution of the mutation across the different cell types. And this is consistent with the fact that ruxolitinib does not eliminate the mutant clones, which is in line with the clinical response, but Rux leads to a symptom reduction, but doesn't reliably reduce clonal burden or even prevents disease progression. So this is showing that even if you inhibit the JAK2, you can actually obtain an even distribution of the mutation across the different cell types, but it does not reduce the overall variant fraction in the patients. So another thing you can do with this method, for example, is to perform intra-cluster comparisons. So here, using gene accessibility as an inference for expression, which has its caveats, but we still see that increased accessibility in the mutant cells correlates with a pro-inflammatory response, which is very well described in myelofibrosis. So it's consistent with what is known, and importantly, it kind of like, since this is an intra-sample comparison, to some extent you can avoid other confounder effects, which are present when you do inter-patient comparisons. But importantly, from ATAC-seq, you can also infer transcription factor motif accessibility. So when we did this differential motif analysis comparison in the HSPC cluster, we saw that there are 2 groups of transcription factors which are involved in pro-inflammatory signaling, such as FOS, JUN, and NF-kappa B, that are highly accessible in the mutant stem cells, while not as accessible in the wild type. So this can be underlying the pro-inflammatory phenotype that we showed before. So as a last approach, we asked whether we can even expand the reach of gotcha to include additional modalities in single-cell sequencing area. So we aim to expand gotcha basically via integration with ASAP-seq. ASAP-seq is ATAC with selected antigen profiling by sequencing. What this allows is to, in addition to using whole cell as an input instead of nuclei, it increases the coverage of the mitochondrial genome, as shown on the circular plot on the right. But it also allows for measuring cell surface protein levels at the same time in the same cell. So by combining gotcha with ASAP, we could have genotypes, increase mitochondrial coverage, chromatin accessibility, and cell surface protein measurements. So we indeed tried this in one sample, and here we can see that we successfully obtained ATAC data, as well as the genotypes captured by gotcha. And in addition to that, we also see increased, we can actually measure protein, cell surface protein levels. As you will notice here, when we perform an ASAP, we notice a slight decrease in the genotyping efficiency, and we think this is due to the requirement of fixation of the cell before the processing. So we think that by fixing the cell, we're somehow impeding the primer binding to some extent. But despite having a reduction in the genotype, the big advantage is that we have increased mitochondrial coverage. So what we did next was to try to find mitochondrial variants that were in phase with the JAK2 mutation. And in one of the patients, we did found 2 different variants that are in phase with JAK2, and that allows us to generate basically a random forest classification model in which we can take these mitochondrial variants and impute the missing genotypes in the remainder of the cells. So when we did this approach, we were actually able to increase our genotyping efficiency for this particular sample to 92% of the cells. So this shows that including mitochondrial information can actually be leveraged for imputation of missing genotypes when using these technologies. So now that we have most of our cells genotypes in the data set, we can ask whether there are any different expression of cell surface protein markers, for example. So what we notice is that in the mutant stem cell clusters, we see an increased expression of CD90, which is actually it's a stem cell marker that is enriched in the earliest stages of stem cells. So when we do a pseudofax gating, we can see that gating in CD34 and CD90 can result in over a 90% enrichment in mutant progenitors. And notably, this enrichment had been reported before in MPN samples, both in bulk and in single cell studies, but always at low throughput. While this data is only from one sample so far, we observe an increased accessibility of the TRY1, which is the gene that encodes for CD90 in these untreated patients. And then we can basically link these epigenetic changes to the increased expression of CD90, which can be used as a phenotypic marker for isolation and or treatment of these mutant cells. So in conclusion, I hope I showed that both GOT and GOTCHA can provide high throughput genotype to phenotype linkage at single cell resolution. And in particular, GOTCHA can surmount the RNA-based genotyping dependencies on gene expression and locus location that we have when applying GOT. We showed also that GOTCHA is compatible with both intact cells and isolated nuclei. And this also amenable to multi-omic integration. So we showed that can synergize with mitochondrial variant calling to further improve genotyping. And it's also compatible with single assays that are dependent of in-droplet PCRs, such as single cell current tag or NTT-seq for measurement of epigenetic modifications. So more broadly, I hope these results show that single cell multi-omic approaches linking genotypes to cellular phenotypes are actually critical for the study of genetic mosaicism. And by moving beyond genotype alone, I hope these methods can provide primary human data to understand how the somatic mutations actually drive these expansions in normal and in normal tissues and how they impact human health. So with that, thank you all for your attention. I want to thank especially your team as well as the collaborators and our funding agencies. I'm happy to take any questions. One last comment, both these works are available in bioarchives, and we have a GitHub R package too if you want to apply GOTCHA. So to basically start to end processing of the genotyping libraries. Thank you.
Great, thank you, Franco. So if anyone has any questions, you can type them now into the Q&A function. So we have one already where Reed Brewer is asking, when relying on DNA for genotyping, is the GOTCHA technology less sensitive than GOT, the genotypes at the transcript level, because there are fewer copies of DNA rather than RNA?
Yes, that's an excellent question. So I would say regarding the sensitivity, it will depend on the target. Basically, what GOTCHA allows you is to be to some extent independent of expression. So if you're aiming to capture the mutation from a target that is very lowly expressed, as I show, your genotyping efficiency can be below 10%, 7%. While with Gotcha, we consistently obtain for diploid organisms between 40 to 60% of recovered cells. Also, what Gotcha allows you to do that you can't do with GOT is to target mutations that are not within transcripts. It could be in any particular region of the genome. That being said, yes, it's true that originally we have only 2 molecules within the drop to detect, and that's why we need to do the exponential amplification. And we actually did a lot of effort into cleaning up basically the noise that arises from this PCR amplification, because the expectation will be that you will have either all wild type or all read, all wild type or all mutant reads when you finish your pipeline. But what we see is actually you have some level of noise within the data, which we think is coming basically from this exponential amplification. It could be coming also from some ambient DNA that is capturing the droplet at the moment. So our package actually includes 2 different pipelines to deal with the noise and trying to correct for it.
Great. And so I also had a question, maybe I misunderstood it. When you switch to the Gotcha method and you're relying on sequencing at the DNA level, do you then, with the GOT method, it's more unbiased in that you're sequencing all the transcripts and you can pick mutations that you see from there. Whereas with the Gotcha, you pick your mutation in advance that you're investigating. Is that the case?
Well, with GOT, you also have to design the primers. Actually, you need to
okay. So in each case,
so in theory, if the gene is very highly expressed and you capture the site regardless of enrichment, should be enough. But that's usually not the case for most
highly expressed.
So for GOT, you still need to spike in specific primers. Yes. So this is targeted. Both approaches are targeted. That's it.
Right. And so with like the specific mutation examples you gave, if you had like a monoclonal population, like if you're looking at these in cell culture, for example, would you have not been able to find the same thing? Like, is there, it seems like one of the benefits might be that you're looking at these in the context of like a broader cell population. Is that one of the benefits of Gotcha?
Oh, I mean, yeah, absolutely. So we did when we do the cell mixing experiments, you can clearly see that the populations are like very homogeneous. Even in the ATAC space, you see that they're clustered very tightly together. So I would say basically Gotcha and GOT, they both have the advantage that you have the single cell resolution of whatever layer of information you're measuring, plus the genotype on top. So this allows you to do like a fine-tuning of the resolution within cell type or within cell states to compare the phenotypic differences between wild type and mutant.
Great, thank you very much. And so it looks like that might be it for the questions today. Thank you again to both Roshni and Franco for the great talks today. Our next seminar will be on October 4th. We look forward to seeing you all there. If you missed any part of the seminars there, if you want to watch them again, you'll be able to find the recordings posted shortly on our YouTube channel. And so thank you again for coming, and thank you again to Franco and Roshni for taking the time to speak today.